right, hopefully third time's a charm because this is the third time that I am trying to record this video. Let's talk about meiosis. So, meiosis is the process that produces gametes. If you've forgotten what gametes are, those are sex cells, aka egg and sperm cells, and these daughter cells are different from the original cell because they are haploid. This is a process that produces haploid cells when your original organisms are diploid. And if you've forgotten what those terms are, don't worry, I'm going to go over them in a few minutes. This is an important step in sexual reproduction because producing those gametes is necessary to reduce the number of chromosomes in a cell before you increase it again to go back to a diploid organism. So, since it's reducing the amount of chromosomes in a cell, this is often called a reduction division. But before we can get into more details about meiosis, let's go over a few background pieces of information. So, in diploid organisms, you can organize chromosomes into pairs of chromosomes that are usually inherited from different parents. You have one maternal copy inherited from the mother and one paternal copy inherited from the father. These contain genes for the same traits in the same order, but they are not necessarily identical because they can contain different alleles for the same genes, but they do contain genes for the same traits. They will also look similar in that the centromere will be in the same location for them, and the lengths of the arms of the chromosome that come off from the centromere, the short arm and the long arm, are going to be the same length. Now, we've talked about haploid and diploid very quickly. Let's go into a little more detail about it. Haploid, you can think of that as standing for half. We think of diploid organisms as kind of the norm just because we are diploid. So, for instance, animals and a lot of plants are diploid, which is going to pop up in just a minute. But side note, when we're talking about the number of sets in a chromosome, we talk about the number n. So n is the number of chromosomes in a set. It's if you only had a haploid cell with one out of each set of chromosome. So the number n is going to be different for different organisms. So n for humans is 23 because we have 23 pairs of homologous chromosomes in our cells. When we have haploid cells, they have 23 chromosomes in them. Our diploid number is 2 times that 23, which is 46. Now, I just said that gametes and fungi are haploid organisms. Well, actually, I talked about animals and plants, but gametes, which are, remember, sex cells, and the multicellular form of fungi are haploid. Animals and some plants are diploid, meaning they have 2 sets of chromosomes, so pairs of homologous chromosomes. Then you have some plants are actually capable of being polyploid, which means they have more than 2 sets of chromosomes. An organism could be triploid, meaning they have 3 sets of chromosomes, but these are usually not going to be organisms that will end up passing on their genes to produce a new species just because of the fact that they will have trouble dividing up their chromosomes during meiosis. So mostly you're going to find even numbers of chromosomes. Like tetraploid organisms are 4n, hexaploid organisms are 6n, octoploid organisms would be 8n. So the ploid refers to a set of chromosomes, and the prefix refers to the number of sets of chromosomes. So let's go into meiosis 1. Before meiosis, I just want to point out, you do have a DNA replication event happen in S phase, where you synthesize a completely new set of DNA. Because of this, each cell is going to have pairs of homologous chromosomes, and each pair of homologous chromosomes, remember, one maternal, one paternal, and they contain genes for the same traits, but they can have different alleles. And then each of those chromosomes is made up of a pair of sister chromatids. Now, the sister chromatids are actually completely genetically identical. They should have the exact same alleles for the exact same genes. Each chromatid after meiosis is going to end up in one of the 4 daughter cells that are produced. So that's where you're basically getting 4 sets of DNA coming from to produce 4 different cells. So meiosis 1 is the first division that happens. It separates homologous chromosomes from each other. This is the actual reduction where you go from diploid to haploid. And at the end, it forms 2 haploid daughter cells. So let's look at each of the stages in meiosis in detail. Just like with mitosis, it does start off with prophase, but since meiosis comes in two different stages, we have a prophase 1 and a prophase 2. During prophase 1, the homologous chromosomes pair up and they also condense. When they pair up, we call this synapsis, where they align with each other and they pair with each other. They'll physically connect. And then when they do this, they form a structure called a tetrad. A tetrad is going to have four bodies, four chromatids joined together, which is where the tetra comes from. Then the meiotic spindle begins to form, the centrosomes move to opposite poles of the cell, and the nuclear membrane breaks down. Again, just like in mitosis, this is a preparing stage. So prophase always prepares. The next stage is metaphase. During metaphase 1, those pairs of homologous chromosomes are going to line up at the equator at the center of the cell, sometimes called the metaphase plate. And again, just like in mitosis, metaphase has them lining up in the middle. Anaphase, just like in mitosis, has the chromosomes moving apart or away from each other. And in this case, you're going to separate homologous chromosomes. So as they've been attached to spindle fibers, and they're going to get pulled apart towards opposite ends of the cell. Notice, however, that sister chromatids are remaining attached to each other. And then telophase, we're going to end with two nuclei. Just like in mitosis, telophase ends with two nuclei. You have the meiotic spindle is going to break down. New nuclear envelopes will form. You'll have some cytokinesis happening at the same time, which means that if it's an animal cell, it'll form a cleavage furrow. Obviously, these that I've depicted here, those are all animal cells. If you have a plant cell, on the other hand, a cell plate will form. Now you have two cells, each of which is haploid, and each of those is going to undergo meiosis II, the second division. This is the division that's going to separate sister chromatids from each other. You're going to start with the haploid cell, and you're going to end with haploid cells. At the end, it's going to form four haploid cells. Each of them is going to have one unduplicated chromosome, and you're going to have two cells formed from each cell that was produced at the end of meiosis I. So what you'll notice is that I have two cells pictured up here undergoing the process because each of these was produced at the end of meiosis I, and they're going to go through meiosis II. So during prophase 2, the meiotic spindle reforms, and now sister chromatids are going to start those pairs of sister chromatids are going to be attaching to the protein spindle. Metaphase 2, again, things are lining up at the middle. Now it's those pairs of sister chromatids lining up at the middle, and you're going to have the centrioles of each sister chromatid is going to attach to a microtubule from the opposite end of the cell. During anaphase, they're going to be pulled apart from each other. So those centromere proteins holding them together are going to break down, they're going to separate, and the sister chromatids will be pulled apart or away from each other to opposite ends of the cell. My bad, I forgot to include the protein spindle here. So these are not just floating chromosomes, they are being pulled towards opposite ends of the cell. During telophase 2, you're going to have the same things that generally happen during a telophase, where the meiotic spindle is going to break down, a new nuclear envelope develops, cytokinesis happens at the same time, again, cleavage furrow in an animal cell, and a cell plate in a plant cell, and then those chromatids are going to start to decondense, meaning they're going to start turning into that stringy chromatin again. So let's go ahead, now that we've gone over meiosis and mitosis, compare the two of them. So both processes use spindle fibers to move chromosomes around. They both are processes to divide up the nuclear contents, so they're dividing up the chromosomes, and each of them has prophase, metaphase, anaphase, and telophase. How they differ is in the number of divisions. Mitosis is one division, meiosis is two divisions. Their products are also different. Mitosis is going to produce cells that are genetically identical to each other and to the parent cell. Meiosis produces cells that are genetically unique, both distinct from the parent cell that they originate from and distinct from each other. Mitosis makes 2 cells, meiosis makes 4 cells. Mitosis is often used to produce somatic cells. You're making new copies, new cells to help a body grow or repair. Meiosis produces gametes. And for mitosis, the ploidy remains the same. What that means is that if you start out with a diploid organism who has 2 N chromosomes, you're going to end with a diploid organism. If you perform mitosis with a haploid cell, you're going to end with a haploid cell. In the meantime, meiosis is going to cut your ploidy in half. So if you start out with a tetraploid cell with 4 N chromosomes, you're going to end with a diploid cell with 2 N chromosomes. If you start with a diploid cell with 2 N chromosomes, after meiosis, it's going to only have N chromosomes, half as many. All right, that is it. Hope these were useful, and we've got another set of notes that'll be coming out soon.